To observe a specific structural failure, we establish a benchtop experiment with 3 separate variables. We pour a liquid biopolymer matrix into 3 glass beakers. We leave the first as a control. Into the second, we add raw tissue from an Ananas camosus, a fresh pineapple. Into the third, we add pineapple tissue that has been thermally processed. This experiment tracks gelatinization, a thermodynamic phase change where a warm biopolymer solution cools down and solidifies into a macroscopic hydrogel. After dropping the ambient temperature overnight, the control matrix and the thermally processed variable have both successfully transitioned into a firm, solid hydrogel. The raw variable remains a liquid solution. This presents a paradox. 2 chemically identical biological additives yield diametrically opposed structural states within the exact same biopolymer matrix. The lack of gelation in the raw sample is the macroscopic result of microscopic mechanical disruption. We can trace this failure to a specific, highly active enzyme present in the raw tissue. This is a structural engineering failure caused by folded molecular machinery acting upon a protein scaffold. To understand the disruption, we must first analyze the native, successful state of the biopolymer matrix. Macroscopic hydrogel formation depends entirely on microscopic protein geometry. Collagen, the foundational structure here, exists as a tightly wound triple helix of long protein chains. The specific sequence of the amino acids glycine, proline, and hydroxyproline allows for the formation of interchain hydrogen bonds, stabilizing the triple helix structure. During manufacturing, applied thermal energy and hydration disrupt these hydrogen bonds. The organized triple helix unravels into disorganized individual polymer chains known as gelatin. As the solution cools and thermal kinetics drop, the individual floating polymer chains attempt to reassociate. They form localized junction zones, where segments of different chains retwist into partial triple helices to anchor themselves together. These scattered junction zones interconnect the long chains, building a 3-dimensional scaffold across the entire volume of the liquid. The negative space within this expansive scaffold permanently traps water molecules, holding them in place. Successful gelation requires polymer chains to remain long enough to cross-link across the solution, establishing the connectivity required for a continuous network. Fresh pineapple tissue contains a high concentration of the enzyme bromelain, a cysteine protease, a molecular machine that locates and severs peptide bonds in proteins. At the enzyme's active site, a single sulfur atom acts as the chemical blade. Through hydrolysis, the enzyme binds to a collagen chain, recruits a water molecule, and cleaves the polymer in 2. In the warm, slightly acidic environment of the initial cooling phase, these kinetics operate at extreme velocity, executing the cleavage reaction thousands of times a second. This reduces the essential long-chain polymers to shortened stubs. This rapid depolymerization occurs in the first critical minutes of cooling, destroying the chains before the drop in temperature can stabilize a network. In the failed beaker, the water pools because the molecular scaffold was shredded before it could assemble. Bromelain prevents gelation by eliminating the chain length required to form junction zones. We can now resolve why the thermally processed pineapple allows the structural matrix to form. Enzymes are proteins, and their function depends on a specific 3-dimensional folded geometry. Thermal denaturation occurs when applied heat permanently unravels the enzyme's folded shape. The high temperatures used in the canning process destroy the spatial alignment of the active site, permanently neutralizing the enzyme's ability to cut the gelatin chains. We can contrast this permanent thermal destruction with the effects of extreme cold, such as introducing frozen raw fruit to the mixture. Extreme cold induces kinetic reduction, slowing molecular velocity to a crawl without altering the protein's folded structure. A thawed enzyme remains intact. It will resume hydrolysis the moment ambient thermal energy rises. Enzyme efficacy is thermodynamically controlled. Heat permanently disables the molecular machinery by destroying its geometry, while cold merely suspends its action. This proteolytic mechanism is found across various biological materials. Homologous cysteine proteases include papain in papaya and actinidin in kiwi. Culinary engineering applies these enzymes to the dense surface of raw mammalian muscle. Tough muscular tissue is a matrix packed with collagen, the same triple helix biopolymer we observed in the gelatin beakers. The food science industry uses these enzymes to manipulate this tough cellular structure. Applied enzymatic hydrolysis shortens the collagen chains in the muscle, reducing its structural integrity. The molecular cleavage that prevented the formation of the biopolymer hydrogel is the same mechanism used to increase tenderness in mammalian tissue. The primary control mechanism is targeted heat, which dictates when hydrolysis begins and when the enzymes are permanently denatured. Our initial beakers show the final physical states dictated by these chemical rules. Food science relies on the predictable behavior of these molecular mechanisms, governed by thermodynamic control and structural geometry.